Yep, it's clear. Please go ahead.
Okay, so I'm going to be talking about epigenomic patterning of cancer cells. And this is the work we have performed at Wayne State University and School of Medicine, Karmanos Cancer Institute. So one of the main goals of our lab is to try to understand how different types of cells express distinct expression programs. So for example, during lineage commitment, how does one cell type express a certain set of cells, a certain set of genes, whereas another expresses a distinct set of genes. So the way to understand that is to think about how DNA is packaged in the nucleus into a chromatin structure, where DNA is wrapped around histones to form a nucleosome. And depending on the compaction of this nucleosome and the chromatin, this leads to differences in the activity of this underlying DNA. So if we focus in and zoom in on a basic repeating unit of chromatin, which is the nucleosome, we can see DNA wrapped around an octamer of histone. And these tails from these histones can be post-translationally modified. So they can be acetylated, methylated, phosphorylated, ubiquitinated, sumolated, and really depending on the modification that's present, this can be correlated with gene activity or inactivity. And this can also facilitate recruitment of different chromatin modifying enzymes, chromatin constituents, transcription factors, etc., which then basically influence the activity state of this underlying chromatin region. So if we think about the genome as a whole, it's basically broken down, compartmentalized into an open euchromatic chromatin region and heterochromatic region. So if we think about just the open chromatin, there are a set of histone modifications present, such as H3K4, histone 3 lysine 4 methylation. And these can be found at open chromatin regions, such as CpG islands, enhancer regions, and promoter regions. And there's also heterochromatic region, which are basically transcriptionally silent regions that contain a different set of repressive histone modifications, such as H3K9 methylation or H4K20 trimethylation. So caveat to this is the presence of a bivalent domain, which was originally identified in embryonic stem cell. And these are basically developmental genes in this case that are poised for activation upon differentiation. And these consist of a kind of a sharp H3K4 active domain that's housed within a broader repressive domain, H3K27. So the thought is that basically these repressed genes can be poised for activation, so they could either resolve to an active state or repressed state following differentiation. So these histone modifications can be found at specific genic features. So I mentioned that H3K4 trimethylation can be found at active promoter regions. And there's also histone modifications that follow the elongation through the transcription of the promoter through the gene body, such as H3K36 trimethylation. And there's also histone modifications that can be found at non-genic region or heterochromatic pericentromere regions, such as these two repressive histone modifications shown on the left. So essentially, if we boil it down, the chromatin is compartmentalized into a silent state that's compact, or a transcriptionally active state that tends to be more open. And there are different features that are associated with these two different states. So DNA methylation is usually transcriptionally repressive. There's also increased density of nucleosomes. There's histone modifications that are specifically enriched at repressive regions such as H3K9, etc. And the density itself of these nucleosomes is higher in a transcriptionally repressed state. Whereas in an open transcriptionally active state, the chromatin tends to be more accessible to binding of TFs and the transcriptional machinery. But beyond that, the actual subnuclear localization of chromatin is associated with activity or inactivity. So if chromatin is associated with the nuclear lamina, it tends to be more transcriptionally silent, more repressed, or if it's more centrally located, it can be more active and transcribed. So chromatin networks reinforce cell states and affect the cell's responsiveness to signals. So for example, in a normal cell, stem cells can transition from one cell state to another because they're able to appropriately respond to specific signaling cues. However, in the case of cancer, where there could be perturbed chromatin networks, they could be locked in a specific state. So there could be an overly restrictive chromatin environment where the cell is not able to appropriately respond to signals. So for example, this may occur if there's decreased levels of active marks. Okay, and then how would you end up with a decreased level of these marks? So this could be influenced by expression of histone-modifying enzymes, for example. And so they're stuck in this proliferative state. They're unable to respond as they would normally in the context of a normal cell. On the other hand, cancer cells may have a permissive chromatin state. So they may be easily transitioned from one cell state to another, some of which may allow these cancer cells to adapt, evade therapies. And this potentially could be due to increased levels of active marks such as H3K4 trimethylation. So if you think about it, say you have an increased H3K4 trimethylation across the genome at promoter region, the cell may be able to kind of adapt by picking a subset of those genes and express those genes to evade therapies and be able to adapt to specific microenvironments. So a lot of this information related to histone modification profiling and transcription factor binding essentially was identified through the use of ChIP-seq, which allows to identify gold protein A DNA interactions. I'm sure most of you are aware of this technology. Essentially, it's basically cells are isolated. The chromatin is cross-linked, typically, and it's fragmented to a mono or dinucleosome level. And then the key is really identifying a high quality antibody that can be used to pull down your protein of interest, which could be a TF, could be histone modification. And then you basically purify this DNA and take it to the library preparation and then perform the downstream bioinformatics analyses. So there's a couple of methods book I highlighted here, which take you through the actual experimental side of the wet lab and all the way through bioinformatics. It's kind of aimed for people that may not have the expertise in both sides. So if you're of interest, take a look at that. So for this study, we were essentially interested in interrogating the epigenomic landscape of a diverse set of cancer cells. So the NCI60 panel represents 9 different types of cancer, 60 cancer cell lines, and there's a lot of different data related to this that's already existing. So RNA-seq, for example, and some other data, but there really hasn't been a comprehensive profiling of epigenetic landscape. So of interest to look at that to understand if there's differences in the landscape between different types of cancer and then between different cell types as well. So what we did is perform ChIP-seq for histone modifications that represent kind of the majority of different active and repressive states in a cell. So H3K4 trimethylation marks active promoters typically. H3K27 acetylation is essentially a proxy for active enhancers. So this allows you to identify intergenic enhancer region. And there's a couple repressive histone modifications, which I mentioned previously, H3K9 trimethylation, H4K20 trimethylation. And what this plot is showing is the number of peaks for these 4 different histone modifications across these 60 cancer cell lines, representing the 9 types of cancer. And you can see varying levels of peaks between these different types of cancer cells. So we wanted to understand the chromatin states of these cells. So we used Chrome-HMM to learn and characterize the chromatin states. Chrome-HMM can integrate multiple types of data such as ChIP-seq. For example, we use histone modification data. It allows you to discover de novo the major combinations of spatial patterns of these different marks. It essentially allows us to annotate the genome across many different types of cells in this case. So in essence, what this method is doing is, on the left you can see representative histone modifications. And essentially what it's doing is it's kind of binarizing these data so that after the genomic bins are established at fixed intervals, it's basically looking for the presence or absence of a specific peak for histone modification to provide this type of data. And then you can learn the model and then provide model segmentation to kind of separate out these different chromatin regions. So using this Chrome-HMM approach, we found we looked at 15 different chromatin states that are present across these cancer cell lines. So on the y-axis is showing these 15 states, and then on the x-axis the 4 histone modifications. And we were able to identify kind of traditional active genes, but beyond that we can identify chromatin states that have different types of enhancer regions. So they could be actually poised or kind of bivalent enhancer regions, meaning they're marked by an H3K27 and a repressive mark as well. And we can kind of break down the genome and look at different types of chromatin states, active, repressed, etc. And that's what these blue kind of squares are indicating there. It's basically the presence of this mark and this specific annotation of this state. So beyond that, we can look at different genic features. So within these 15 states, we can also look at the representation of CPGs. We can look at different features as well, such as exon genes, etc., which are indicated on this x-axis. And we can also look at general transcription or activity of genes within these 15 states. So, for example, we can see CPGs are enriched in these states shown here, which most likely indicates that there's an greater presence of CPG islands in these specific chromatin states. And then we can also look at relative genome coverage. So across these 15 states, we can see the relative coverage for each of these 9 types of cancers. So then beyond the initial chromatin state mapping, we can look at essentially higher order chromatin associations across these 60 cancer cell lines. So this is a kind of a semi-complicated plot, but essentially what we're looking at is, again, it's the 15 states on the x-axis, but the y-axis is basically looking at 2 megabase genomic bins. So across the whole genome, we kind of broken down the genome into these 2 megabase intervals, and then looked at within these 2 megabase bins, what is the relative representation of these states in these regions. So this gives us more information as far as correlates between gene density, etc., and also relative to cytogenic banding. So it gives us further dimensional information into this chromatin state information. So we also looked at the presence of specific repeats in these 15 different states. One of the reasons for doing so is because derepression of repetitive DNAs can be correlated with the dysregulated chromatin or chromosome aberrations in cancer cells. So for example, we can look for enrichment of line or LTR retrotransposons, which may be derepressed in specific cancer types. And this gives us more information as far as the enrichment of these repeats across these states and also repeat family members across these 15 different states. Another interesting thing we found is there's differences in chromatin state switching across these different cancer cell lines. So why is this important? So again, I mentioned that chromatin across the genome is basically broken down into an active and inactive chromatin state. So if we can identify differences in the transition between an active state and an inactive state, this may provide some insight into the dysregulation of a chromatin landscape in cancer cells. So we found some interesting results related to the switching from one state to another across these different cancer cells. So for example, this I'm not going to go into all the detail, but basically this is showing the switch from this specific state to another state. And so there may be different features related to enhancer activity or promoter activity or repressed chromatin, which may end up spreading from one state to another in one cancer versus another, or from one cell type to another. We also looked at essentially active promoter profiling through H3K4 trimethylation profiling. And this principal component analysis really demonstrates that there's varying heterogeneity between different cancer types. So we can see the green triangle represents the colon cancer, which tends to be more clustered together relative to, for example, breast cancer, which tends to be more scattered. So this could indicate maybe there's potentially more heterogeneity in some cancer types versus another. And this will allow us to look at specific differences between cell types within a cancer, within a specific type of cancer. On the bottom here is showing the annotation of these peaks for H3K4 trimethylation. Essentially, where are these peaks located throughout the genome relative to a genic feature or a chromosome region. So essentially, we're seeing that these are mainly found in promoter, intron region, and intergenic region. So we can see there are some differences between the cell types related to the overall enrichment. Another interesting thing we observe is the similarity in the actual peaks between these 60 different cancer cell lines. So this is basically showing a pairwise intersection of these peaks across these different 60 different cancer cell lines. And functional annotation of these peaks may also provide some insight into what types of genes may be patterned or occupied by these histone modifications. So just going to mention one specific thing. So we can look at all of these peaks and annotate them all. And we can also look at cancer-specific peaks. So these are unique peaks to a specific cancer type. And one thing I'll mention is we see enrichment of developmental genes. If we look at unique genes for each of these cancer types, suggesting there could be some potential developmental genes that may be de-repressed, activated in different types of cancer cells. So, okay, so we also looked at basically essentially the mutation analysis of these occupied regions to see kind of mutation burden of these cancer cell lines. So this plot is just showing the overall mutation count across these 60 cancer cell lines. And we wanted to understand if there's either an increase or decrease, or what exactly is the mutation density of these H3K4 trimethylation peaks relative to other genomic regions. So we saw kind of a significant increase in mutation density in regions occupied by H3K4 trimethylation relative to random regions. Okay, so this is basically the similar size and frequency of these peaks, but also relative to regions without H3K4 trimethylation and random regions. So this basically means that there's increased mutations in open chromatin region, which kind of makes sense because if you have a decondensed chromatin, it may be more susceptible to breaks or some other mutation during DNA replication or during transcription. So keeping a compact chromatin state tends to kind of lock it in more of a stable state potentially. Okay, so we also looked at the patterning of H3K27 acetylation, which essentially is a proxy for active enhancer. And if we look at the overall pairwise intersection between these different cancer cell lines, we can see there's kind of a significant overlap of the melanoma cancer cell lines and also some renal cancer cell lines. I'm not going to necessarily go into this as much. This is kind of the general annotation, which I mentioned before, but I will mention that as we found for the H3K4 trimethylation, similarly, we see increased mutation density in H3K27 acetylation marked regions relative to random region and the rest of the genome and then the rest of random genome. The difference was not as significant. It was still significant based on p-value. So it further suggests that open chromatin tends to have more mutations than closed or compact chromatin. Okay, so this previous plot is basically showing all of the peaks for H3K27, all the intergenic H3K27 peaks. We also looked for cancer type specific H3K27 intergenic enhancers. And surprisingly, we also find that there's really a larger or greater number of melanoma specific intergenic enhancers that are cancer type specific relative to the other 8 types of cancer cell lines. So this is basically the cancer type specific enhancers, and it's just sorted in ascending order from breast cancer all the way through renal cancer. So beyond typical enhancers, which I mentioned, there's also super enhancers, which folks are probably familiar with. And these are clusters of enhancers within a 12.5 kb region. And these are more cell type specific. So they're essentially defining cell states. So we identified these from the super slope plot. So these are going to be these points above the region where the slope is around 1. So basically these regions. And if we cluster these super enhancers shown here, we can see that these are basically cell type specific. There's not really an overlap of super enhancers between each of these cell types. So these can be used to really identify individual cells. And these potentially could be used then to categorize or diagnose, etc., patient-specific tumors. So it could be used as kind of leverage for developing some personalized type of therapy or diagnostic. So we see there's actually an increased number of superenhancer in cancer relative to the normal tumor, normal tissue. So we can see a greater number of superenhancers breast cancer versus a normal memory tissue. And this can be said for basically majorities, except for I think maybe this one, because it's not, they're all kind of clumped together. So it's a little more difficult to separate them out. We also see the increased activity of superenhancers for the majority of these cancer types. There's a caveat to that. So CNS is actually a slightly decrease. In renal cancer, slightly decreased as well. But for the other ones, we see the activity of superenhancers tended to be increased in the cancer cell relative to the normal. So another interesting feature we found is there's enrichment of specific consensus binding motifs for transcription factors in these superenhancer regions. So what this graph is showing is these are transcription factor motifs that we identified in superenhancer regions across these 9 different cancer types. Okay, so the TF is on the y-axis, the cancer type is on the x-axis, and what each of these circles is representing is the expression of a TF in that specific cancer type or cell line is indicated by the green to red coloring. So if it's red, it means that it's expressed at a higher level, that specific transcription factor. So there's a correlation between the actual expression of the TF and the motif being present in that superenhancer region. And then the size of that circle indicates the significance of that specific motif. So basically, if it's a large red circle, it means that there's more significance applied to that TF being expressed and the motif present in enhancer region, that superenhancer region. So we found an interesting set of TFs that are present in a cell type and in cancer type specific manner as well. And these could be potentially exploited to, again, develop some personalized therapies or for diagnostic purposes down the road. Okay, so I'm just going to mention, so beyond the active profiling of H3K4 trimethylation and H3K27, which mark the promoter and enhancer region respectively, we also looked at heterochromatin profiling, essentially looking at repressed chromatin. The thought being that if you have dysregulation of heterochromatin, this can kind of wreak havoc on chromosome stability, lead to chromosome aberrations, etc. So we looked at these two, which are kind of the most studied and most correlated with the repressed chromatin. And we found it's actually greater heterogeneity of these marks relative to the other active marks, meaning that you could have more dysregulation of the actual compact chromatin relative to the active chromatin. This is showing just principal component analysis of that result. This is just much more scattered relative to the other 2 active marks. And again, this is mainly annotation information, which I'm not going to discuss, but I will mention that the mutation density is actually lower in regions that contain H3K9 or H3K20 relative to random region or basically the rest of the genome without that specific mark. So this is opposite of what we found for the H3K4 trimethylation and the enhancer activity mark. So just in the compact chromatin, these specific regions tended to have a decrease of those specific like SNP mutations and those type of things.